Structure of DNA. DNA molecules are long. Human genomes have about 3 billion bases and about 20,000 genes. Each molecule of DNA is about double helix made from two complementary strands of nucleides held together by hydrogen connections. The genetic information is carried in the linear sequence of nucleotides in the DNA. DNA function. DNA contains biological and genetic information that is controlled day to day. DNA is like a blueprint that contains the directions necessary to construct other cells. For example, proteins and RNA directions are passed down to create inheritance. These are the DNA parts labeled in this picture right here. DNA replication is the process of producing two identical replicas from one original DNA molecule. This process causes in all living organisms and is the basis for biological inheritance. DNA helices binds the DNA. Helices are enzymes that bind and can even remodel nucleic acids. There are RNA and DNA helicases. DNA helicases are essential during DNA replication because they separate double-stranded DNA to single ones, allowing each strand to be copied. In this picture, the cell cycle is explained right here in this slide. DNA polymers makes a new DNA by adding more complementary nucleides to a strand. This picture below explains how the DNA polymers is made. DNA being copied. The point where the double helix opened up and the DNA is being copied is called replication fork. Once the strands are separated, an enzyme called DNA polymers copies each strand using the base pairing rule. The two strands are not exactly copied in the same way. S phase of the cell cycle is a part of the cell cycle in which the DNA is replicated. Causing between G1 phase and G2 phase, precise and accurate DNA replication is to prevent genetic conflicts which often lead to cell death or disease. Transcription and translation is the process by which the info in the DNA is copied into the messenger RNA and mRNA for protein production.